This is the third video on biodiversity. So we're looking at how we measure biodiversity. We've just looked at community and how we measure the biodiversity within these interacting populations of organisms. So now we're going to look at individual populations and how would we measure biodiversity there? Because we can't use species richness. That only applies at that community level. How many different species do you have? However, populations aren't going to be applicable for that measurement. So hopefully you'll remember that a population refers to individuals of the same species interacting together in the same place at the same time. So we can't use species richness because that number is always going to be 1 if we're looking at populations. So we need to look at other metrics. One of those is morphological differences. So within a population, we will have variation. And some of those will be morphological. We'll be able to look at the physical, observable differences between those organisms. And here are some examples. So these are each pictures of the same species, so a population, and within that population, the variation. So we can see some moth wings with variation in their coloration. We can see bunnies as well with variation in their coloration. And there would be other morphological differences, but sometimes these are the easiest to see as color. So if we're going to kind of focus on this one in the middle. An inadvertent experiment demonstrating genetic variation within a population, courtesy of my daughter. 2 varieties of radish, one long, large, and white, the daikon, and the other round, small, and red, the German giant, were planted in adjacent beds and allowed to go to seed. This is the result approximately 1 year later. So notice this is within a population. So all of these radishes are the same species. And natural populations probably looked something like this, where you had a variety of different features, some more bulbous, some longer, some more bulbous with a longer, thinner taproot attached to it, some more purple, some more white. Right? We have this morphological variation within these species. And then we as humans like to tinker with stuff, so we wanted to have radishes that were rounder and smaller and redder. So we kept selecting ones that looked more like these two and breeding those together and letting their genes recombine to then hopefully make our way toward eventually getting this German giant. Other populations maybe thought that the white radish were more delicious looking. I don't know. And the longer they were, maybe they liked the sort of evenness of them because it made them easier to chop. I have no idea. But eventually, by selecting for longer and less pigmented varieties and breeding those together, you could get the daikon. So these are just 2 extremes of the natural variation that can exist within this population. If you breed together ones that are similar to each other and then kind of get increases in all of those phenotypes. So here we have this demonstration of these being the same species interbreeding with each other and their offspring showing that variation that exists within these natural communities. And we have done this with many different types of plants. One that you might be the most familiar with is Brassica oleracea, and this is has a general name of wild mustard, and you've probably seen it out growing around. And there's other ones that look like it too, like the one that you'll probably see that looks like this there's a wild radish and a wild mustard, very closely related, both brassicas. However, you might be less familiar or less associate this with the things that you're more familiar with. Let's look at them. So this particular plant has been artificially bred and undergone selection for different parts. So if we were to select specifically for large buds where it would be making these sort of lateral buds, we could get Brussels sprouts. If we were to select for a large terminal bud, so the bud at the very end, we would get cabbage. If we were to select for the flower buds having this very particular weird morphology, we could get cauliflower. And you just keep breeding ones together. So let's say this particular individual has very large leaves, and you want to eat those leaves. So you breed it with one of the other ones in your garden that has large leaves as well. So you would take the pollen from one and deposit it onto the other and hope that those large leaf genes were given to the offspring, right? And that there's some synergistic effect that increases the size of those leaves in the offspring. And over time, you just keep breeding those offspring together and you get larger and larger leaves and you get something like kale. So all of these vegetables that you're familiar with eating are all the same species. We've just selected for different parts of that plant. And none of them look anything like this wild variety. So morphological differences are one way that we can measure biodiversity within a population. Another way is that we can look at genetic differences, and this tends to be the primary mode of study now. So genetic differences refer to the differences in the order of nucleotides in the same region of a chromosome. So some of that might be new terms for you, so we are going to look at those. First thing that would be important to talk about is what is a chromosome? Chromosomes are tightly packed bundles of DNA. So inside each of your cells of your body, your trillion cells, you have 6 feet of DNA inside the nucleus of those cells. 6 feet of linear DNA. A cell is very small. So to fit all that DNA in an organized fashion into your cells, they are wrapped around proteins, supercoiled, and then condensed into this package called a chromosome. And that's what we see here, just a super condensed packet of DNA that allows us to organize and store DNA so that when we divide our cells to replicate them, it's easier to not mess up. In your body, in all of your cells except for your gametes, so if you're making eggs or sperm, you're going to have 46 chromosomes. And each of those has a pair. So there are only 23 kind of different versions of chromosomes. And for each of those, there'll be one that you got from your first parent and one that you got from your second parent. So this one maybe I got from my dad, and this one I got from my mom. And that goes for every single one of these different chromosomes. And each of those pairs of chromosomes will code for different genes. So genes are these little kind of functional units. Imagine that your DNA is a giant instruction manual for how to build you, and that each chromosome is a different chapter in that instruction manual. And within that chapter, you'll have many different sets of instructions for all sorts of different things. When I teach about DNA and proteins in biology, I tend to think of this as the IKEA. So your whole body is an IKEA, right? And you have all of these different rooms, and you have instructions for how to build everything in each of those different rooms. So each little instruction manual for a different part is coded for by a gene. And those genes are within these sort of larger organized regions called chromosomes. So you would have genes that coded for important things that determine who you are, and you would have the same genes on each of these chromosome 1s, but you might have different versions of those genes because maybe, my dad had blue eyes and my mom had green eyes. So I inherited a blue-eyed gene from my dad and maybe a green-eyed gene from my mom. That's the same gene though, it codes for eye color, right? And there's multiple genes that contribute to your eye color, but we like to simplify it and just say there's one. So same gene, different versions. Those different versions are called alleles, which is allele. It's weird to say. So in a population of organisms, this is just me. This is, my particular karyotype. This is what my chromosomes look like. Not really though, because this would make me a man, biologically. So I have my sets of alleles, my different versions of genes, and that is highly dependent on who my parents were and what their sets of those different alleles were. I will differ from my siblings slightly because we each had this kind of random combination of different alleles from our parents, but we'll also be more similar to each other likely than we would be to other people that we know who aren't directly related to us. And if we look at the entire human population, we might have many different alleles for the same gene, whereas if we look within a family, we might only have the potential to have 2 or 3 different alleles, or maybe even only 1. So this is a little bit of a summary from what we just talked about, but each of these chromosomes has a different set of information. And you can think of them as chapters to the instruction manual that is you. Everything in your body, even your behaviors, a lot of those are determined by your genetics, right? And that's something that we're still exploring. But we have this code within us that determines the types of products that will be made in our cells, that determine how we interact and respond to our environment. So how do we measure genetic diversity? Here we're looking at a mouse gene and a fly gene. So these are the same gene, and we have mostly identical sequences. So each of these letters is called a nucleotide. So we have A, T, C, and G. Those are our 4 options for nucleotides in our DNA. The synthetic DNA that people are building, they're trying to put in extra nucleotides and see if there are different types of proteins that would be made. Very interesting research. So here we can see that the areas highlighted in green are the same. So looking at a gene from a mouse and a gene from a fly that are very distantly related, they still have the same nucleotides in the same order. So that tells us that these 2 organisms likely descended, potentially, from the same common ancestor. So there was a gene back in the day, back in some organism, that had this same sequence. However, over time, as DNA gets replicated, there's mistakes. Sometimes those mistakes provide functions that are better suited to the environment that you're in and allow you to survive and reproduce better. Sometimes those mistakes cause you to die because they were less beneficial. They lowered your fitness, your ability to survive and reproduce. And sometimes they're completely invisible to the kind of products. So if you mutate certain nucleotides, if you get a difference in those nucleotides, if they're in a particular position, it won't change the protein that's made. So really the only way to tell that there was a mutation would be to read the DNA. Other mutations might cause you to not be able to make certain products, like maybe you can't make insulin, so then you have to get insulin injections to be able to live a normal life, right? So vastly different outcomes. You could also get a mutation in your gene that makes codes for the products for your earwax, right? And you get a single nucleotide mutation there, and suddenly you have crusty earwax instead of waxy, kind of wet earwax. It's not definitely going to contribute to your survival and reproduction, but we can still use it as a trait that organisms have, or that humans have, to kind of tell heritage and who you're related to. Anyway, I went on a long tangent there. What we're looking at are these differences. So as your DNA gets replicated and you make new cells and you make new organisms, there is the potential for mistakes, and those mistakes tend to accumulate over time as long as they don't kill you. And so the farther in evolutionary time we get from the relationship where these 2 shared a common ancestor, the more changes they're likely to accumulate because mutations happen at a predictable rate. So each of these mutations is called a SNP, and you can just remember the term SNP, but SNP stands for single, so 1, nucleotide, one of these A, T, C's, and G's, polymorphism, so that's like morph, you're thinking the form, so it's a polymorphism, there's a difference in form there, and there's a couple different options for you. So it's a single nucleotide polymorphism. We have a difference at that single nucleotide, so it's a SNP. So we can look at the accumulation of SNPs in genes that are present in a large group of organisms. So this one, cytochrome C, is involved in cellular respiration, and from unicellular to multicellular, most of the organisms that are eukaryotic are going to have this process So cellular respiration is how we get energy from our food. It's the reason why you breathe in oxygen and breathe out CO2. So cytochrome C is just a piece of that machine. So the gene that codes for cytochrome C, if anything goes wrong with it, if there's a mutation that makes it in any way change its function, that could potentially lead to you not existing at all, right? Because that is an essential process that all of your cells need to go through constantly. So mutations to cytochrome C are relatively rare because you have to have basically silent mutations, these ones that don't change the product, for it to keep functioning the way that it does. So we have a low rate of mutations in this particular gene, a low rate of SNPs. Otherwise, if we were to look at a gene that isn't as important, we might get too many mutations to be able to say anything about these really distantly related groups. So we can look at and kind of have this predictive rate for how often we're going to get a mutation in our cytochrome C, and we can use those and look at the differences in the genes for all of these different groups of organisms, right? And Saccharomyces. So looking at the differences in this gene in all of these different groups and saying how different are they from each other? Because over time you are likely to accumulate mutations in this gene at a particular rate. So if we have a lot of differences, then we can calculate backward when the likely divergence time was for us and something like yeast. Or we can say if there are very few differences or no differences, that we would be very closely related to that organism because we would expect to see mutations accumulate at our expected rate. Just because you expect something doesn't mean that it'll happen, right? It's a matter of probability, but we can put together a likely hypothesis for how closely related all of these organisms are based on that mutation rate. So how can we use genetic sequencing? That's one way, looking at and trying to piece apart our evolutionary histories. Often there's too many changes or the changes kind of contradict each other in different genes to be able to say how an evolutionary history occurred. Cytochrome C is a particularly valuable one, but only for distantly related groups. We couldn't take genes for cytochrome C within the human population and use them to tell who's closely related because they would all be too different from each other. So here we have my study fungus from grad school. This is Heterobasidion, and it used to be all called Heterobasidion anosum. But eventually they figured out that if you tried to breed different ones together, they didn't breed. So the biological species concept is this idea that organisms of the same species can successfully produce offspring together and that organisms that are not the same species can't. So figuring out that some of these couldn't reproduce with each other indicated that they might be different species. And this is what that information looks like. So I did a lot of DNA extraction and then sent it over to sequencing and got that information back and put it into a program. And the best thing about doing genetics in today's world is that there are programs that make it look very beautiful and easy to understand. So all of these gray bars, if we were to have a closer view of this, the computer would report them as the different nucleotides. And those gray regions represent no difference from whatever I've chosen as my kind of reference sequence. So this reference sequence, anything below it that's still gray is identical to that nucleotide position. So anytime we have one of these colorful bars, that represents a nucleotide that is different from the reference sequence. And you can see some patterns forming here. Almost all of these are identical for the whole sequence to the reference sequence. So we would call those all the same species. This one happens to have a mutation, but mutations happen, and that doesn't make it a different species if we just have one. This one here has kind of a lot of SNPs from our reference sequence, and all of those are shared with each other. So we have the same set of SNPs happening in this group. So this one is Heterobasidion occidentale, and this one is Heterobasidion irregulare. And it's really difficult to say what a species is. So because these two couldn't breed together, they were called different species. And we can look at their genetics and figure out who belongs to which species, because it's really difficult to do it morphologically. They don't have a lot of differences visibly, but they do infect different hosts. So you're more likely to find occidentale on firs or spruce, and you're more likely to find irregulare on pine. So those usually occur in different habitats. It keeps them kind of separated, and we get these accumulated differences because they're not mating with each other, mixing those genes together. So we get distinct sets of differences where we can visibly see the difference between their sequences because of this program. Interestingly, this was what I was studying. So we see a SNP here that matches irregulare, and a SNP here that matches irregulare, and one here. Then we have several SNPs that don't match anybody, and here we would see regions that match occidentale, where it's gray, where we have differences in heterobasidion. So all of these represent similarities to the other species, right? So we have a mix. We have a mix of mutations from one species, and these would also technically kind of be mutations from the other. And then all of this is the shared stuff, right? That's still the same, it's conserved. So I've drawn a bunch of confusing looking stuff on here, but what I want to show is that, I'll just erase all this so we can look at it. This mixture of these different SNPs gives us some idea that we had some hybridization between these two species. So we found both of these species in a particular area in Montana, and the hybrid species, or the thing that we thought was a hybrid species, was parasitizing a totally different tree than either of the original parent species. So this was on larch. So that led to the question of who was it? What species was now able to infect larch? And it appears that it's a hybrid between these two things that we thought couldn't breed with each other. So that is part of how we use genetic sequencing.